every living thing has DNA, we're actually quite similar DNA-wise to animals and even plants. 50% of our DNA is the exact same as the DNA in bananas, which is why I'm wearing yellow today. So in this video, what we're going to be doing is we're going to be taking DNA out of strawberries, which is really, really cool. So DNA stands for deoxyribonucleic acid. Every human, every plant, every living thing is made of cells, and inside of each and every cell is DNA, which is like the instructions for how everything's supposed to be and what it's supposed to look like. So what we're going to do is we're going to try and take some of that DNA out of some cells and have a look at it. The thing about DNA is it is super duper 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 tiny. It's nano size, so we can't actually see it with the naked eye. It's super hard to see. But what we can do is if we can clump a load of it together, so we have like thousands and thousands of them in a big bundle, then we can actually see it with the naked eye. So that's what we're going to try to do. We're going to use strawberries today because strawberries actually have a surprising amount of DNA in their cells, much more than, we'll say, a human cheek cell, for example, which is often used. Strawberries are packed filled with DNA. So that's what we're going to do. Okay, so you need a few different bits and bobs. The first thing you need is obviously strawberries. Then you need what we're going to call a DNA extraction fluid, and that sounds fancy, but it's really not. It is basically water, salt, and washing up liquid, or detergent, whatever you call it, and I'll show you how to make that in a minute. You'll need Ziploc bags. You need a filter. You need one way of separating a solid yuck from a liquid, and the best thing I have found to use are coffee filters, which I will show you in a little second. They work really, really well, and they're much cheaper than any fancy thing you might get your hands on. And then the last thing that you're going to need is alcohol. And this isn't like drinking alcohol. This would be high concentrated ethanol. So if you want to get your hands on some alcohol without raiding a lab, you can get rubbing alcohol, or as we call it in Ireland, surgical spirits, which you can get in a pharmacy. If you go in and ask them for surgical spirits, you can get your hands on some. And what you need to do with that first, before you start the experiment, is have that chilling in a freezer. If you're doing this somewhere without a freezer, you can just put a load of ice around the bottle and that'll cool it down. Okay, so first of all, we should get started with our strawberry. So what we want to do first, use our Ziploc bags. We're going to get a nice big strawberry, 1 or 2 strawberries, and take the green bits off of it. We don't need them. So we're going to pop them back in. And we're going to pop these strawberries into our bag. So as I said, the DNA is inside of the cells in the strawberry. But what we want to do is we want to expose as many cells as possible, basically. And so to do that, we get to crush up our strawberries. So that's the best part. So we can start squishing some strawberries. These are hard ones. I should have picked squishier ones. So if you get yourself, you can get strawberries that are almost gone off, and they'll work as well. They might be easier squished than the ones that I've managed to pick. Sorry if this is not usable. So we want to squish it up as much as we possibly can. Do not sit it on the table and slam your hand off of it. Don't try and like bash it or anything like that. You don't want to pop the bag and be covered in a load of strawberry gunk, okay? So squish it up as much as you can. It can be quite nice. So yeah, so it's kind of like this, or squishier. And the next thing we have to do is add our DNA extraction fluid. For that, you need half a cup of water, about 2 teaspoons of washing up liquid, and a teaspoon of salt. It's not super accurate or set in stone or anything, so if you go over or under on that, it's not really a big deal. The important point is having all of these things in here. That's about a teaspoon. More. Yeah. Sure, grand. Okay. I know how to measure things. It's grand. Okay. So you want to mix it up into the water. Obviously, it's filled with washing up liquid, so you don't want to make a load of bubbles, so just try and do this slowly and gently. So what you're going to do next, once this is mixed up, is you're going to put some of it into your mushy bag of strawberries, which we can still be squishing. So these very important ingredients, both play a role. So now that we have squished up the strawberries, loads of cells have been exposed basically. Cells, especially plant cells, they have different kind of structures keeping everything inside, shall we say. There's cell membranes and there's cell walls. And what we need to do is we need to burst open the cell walls to release the DNA that is inside. And to do that, that's washing up liquid's job, basically does that. So washing up liquid is really, really cool. It is washing up liquid, as you know, if you've ever done the dishes at home or been useful around the house, is it dissolves dirt and oil and grease and stuff like that. And it dissolves it away into water so you can't actually really see it. And it does that by having two main parts. One part that loves water and one part that kind of hates water but likes oil. So what it can do is it wraps itself around oil and that helps it dissolve into water and go away. So the same process is going to happen here. It's going to attach to kind of the parts of the cell that we want to burst open and help that be dissolved into the water and burst it open, basically. I hope that made sense. The job of the salt then is to help all of the DNA strands, which are now floating around in the water, help them all clump together. So the DNA clumps all the DNA strands, sorry, the salt clumps all the DNA strands together. So now that this has a nice, that's holding it up far too high. So now that this has this squirty top, is much easier for wee kiddies and things to pour their liquid in. You can just do it in a cup or anything like that. But so what we're just going to do is we're going to squirt some of this in here. Now, if you use a nice washing up liquid, this probably smells really, really nice right now, especially with the strawberries. So I'm going to stop you right now and tell you, don't eat this. It might smell nice, but don't eat it. Okay, so we're going to squish this up a bit more, let everything mix around, make sure that the washing up liquid is helping burst the DNA out of the cells, and the salt is helping then clump up the DNA together. Okay, now that that's been done, even though you can't see it, there's loads of DNA kind of floating around in here, but it's also mixed up with big thick lumps of strawberry that we have no use or need for right now. So what we're going to do is we're going to try and separate them out. And that's where our coffee filter comes in. So coffee filters have a beautiful design. That means you can just sit them into cups and glasses. Ta-da! And requires a little work and effort on our part. You don't need fancy funnels or anything like that. Okay, so this should be okay. I hope so. Okay. Yeah, it does smell really good. Okay. So we're going to hold this and pour it in. And gorge. Yeah. So what you'll start to see after a minute, just going to make this sit here so I can have as little job to do as possible. You'll start to see the liquid dribble through, which is exactly what we want. Now, do not be tempted to just grab a hold of the top and squish and just force the liquid through, because you will burst the bag and have to start the filtering all over again. So we'll let that drip through. Okie dokie, so we've been waiting here a wee little while and some is dribbling through. If you don't have coffee filters, by the way, you can do this with like a sieve. You will still get some kind of lumps in it, but it will take like the bulk of the lumpy strawberries out of it. So I think we should have enough now, and I'm not going to risk it by squeezing it again. So we're just going to take this filter paper off and we can tidy our mess into our Ziploc bag that we already used. Okay, so obviously if you waited longer, you'll get more liquid out, but we should have some DNA here. Okay. Believe it or not, there is DNA swirling around in here. You just cannot see it yet because DNA is soluble in water. So soluble means that when you put it into something, it appears to disappear, although it is actually still there. So if you think of putting sugar into water, sugar can disappear, but if you were to taste it, you would still taste the sugar. It still is in there, you just can't quite see it. So DNA is in there, we just can't see it. So what we need to do is we need to put something in there that DNA does not dissolve in, that it appears in basically. So that is our ethanol. Okay. Now, super chilled in my fancy science lab bottle, which you don't need. Oh God, it is very cold. Okay. So I am going to do this with a dropper, but you can pour this very, very gently in with just going very, very slowly at home. So what we don't want is we don't want to like throw a load of this in really, really fast and mix everything up together. What we want is we want to try and slowly add it on top so that it kind of creates a layer. So we're just going to dribble some down like this. And then after a while we'll just start seeing like a clear alcohol layer on top. Now, so unfortunately what we won't see is we won't see the lovely helix structure of DNA. We won't see that up close, but something will appear in the glass that wasn't there a few minutes ago, and that will be our, well, that was there, we just couldn't see it, and that will be our DNA. And it is kind of gross looking, which is why I think it's super cool. I think it looks kind of like spit. Okay, so some is starting to appear here now. If you have a wee little look in, it almost kind of looks, as I said, kind of like bubbles, but really like spit. So if at this stage, if you'd like a tweezers or like a little stick or something, you can take it out. So I'm going to get a wee lollipop-y stick thingy. And I'm going to try and collect some up and pull it out. So you know this isn't some of the solids from the strawberries because you've very diligently filtered them all the way. But here's some of our DNA. Like spit. It's really gross. It's great. Okay, let me collect up a bit more. Ew. It's so cool. Okay, it's real gross and real cool. And you can collect this if you want to keep it. You can keep it in like a tiny little jar, like a sealed jar with the alcohol in it, and it should last for a while. But yeah, that's our DNA that we have successfully harvested from our strawberries. And you can use similar methods to take DNA out of pretty much anything. But obviously, if you need to use them for like forensic purposes or anything that might be a little bit more scientific than a Ziploc bag. And that is how you extract DNA from a strawberry. Spoiler alert, I always squish the bag. Shh, don't tell the kiddies. I always squish the bag, but I'm a pro. Oh no! Ooh.